get started. Thank you everyone for joining. Really excited to kick off our 2024 Translate series with Dr. Dan Bauer. For those who aren't familiar, Translate is a series that the clinical operations team at Nucleate, which is a student-run sort of biotech interest and development group, started last year as an effort to get clinicians and clinical trainees more involved in efforts in pushing forward therapeutics and diagnostics in the form of biotech companies. This year, our series is focused specifically on understanding emerging biotech innovations in the clinic. So technologies and new therapeutic modalities that are just on the cusp of reaching broader clinical adoption, and what it'll sort of take to get those things from in the lab to in these sort of earlier stages of clinical development to really being used at scale to treat patients. So I guess a quick intro for those who don't know me. My name is Shia Wedja. I'm a MD.PhD student at Harvard Medical School and MIT, and I'm also the co-vice president of the clinical operations team here at Nucleate. And I'm joined by Jessica. Maybe Jessica, you could give a quick introduction.
Yeah, hi everyone, I'm Jessica. Thankfully, also in the program with Shia, that's how I met him. And he brought me on for this amazing talk today.
And so without further ado, let's get to our speaker here. And our first session focused on genetic medicines for inherited blood disorders. We're joined by Dr. Daniel Bauer. Dr. Bauer is the director of the gene therapy program at Boston Children's Hospital and an attending physician at the Dana-Farber Boston Children's Cancer and Blood Disorders Center. He's also a leading expert in genetic medicines for inherited blood disorders. And his postdoc work led to the identification of a previously unidentified genetic element that was crucial for the regulation of fetal hemoglobin expression, which is a discovery that directly enabled a number of gene editing therapies for hemoglobinopathies, including a few that he'll describe today. His research laboratory at Boston Children's Hospital continues to investigate genetic determinants of blood cell development and disease to enable novel treatment strategies for blood disorders. And with all of that expertise, we're very excited to hear his insights on this topic. So without further ado, we'll pass it on to Dan.
Fantastic. Let's see. All right. Can you see and hear me okay?
Yep, we're all good.
All right. Well, yeah, Shia, thank you for having me here. Awesome to participate in this student-led effort, and really an exciting time, I think, to talk about genetic medicine for blood disorders, and we'll focus on sickle cell disease as the most common inherited blood disorder, and really the example that will be the through line throughout. So we're kind of at this period of mixed emotions. There's a lot of excitement. We have ex vivo gene therapies where we can find patients who have an appropriate clinical status, who want to participate, mobilize and collect cells that can be edited or modified outside the body. And then those genetically modified cells might engraft after something like myeloablative conditioning therapy. And then we can follow patients and hopefully have outstanding outcomes. On the other hand, we're faced with the reality that there's lots and lots of patients with sickle cell disease. The right shows a cartogram of the incidence of sickle cell disease, where the area of a country is scaled to the number of newborns expected to be born with sickle cell disease over the coming decades. And you can see that although we have 100,000 patients in the United States, it's really kind of a drop in the global bucket. Most patients are in Africa and in India, and the current therapies are just really not scalable to meet the clinical unmet need. So I think that really motivates us to do more and do better. Here are my disclosures. So I'm going to start with what I think is really an astute and prescient clinical observation that was made by a pediatrician, Janet Watson, working in New York in the 1940s, who figured out essentially that fetal hemoglobin is the main modifier of sickle cell disease. And just remember, this is before the Linus Pauling had described sickle cell disease as a molecular disease, before the structural hemoglobin had been described, before the knowledge of the genetic mutation that causes sickle cell disease was understood. And so Dr. Watson writes, the theory that it once presents itself is that fetal hemoglobin is unable to produce sickling, and the sickling trait progressively becomes 100% with the gradual formation of the new red cells containing the adult type of hemoglobin, which possess the sickling property. This hypothesis is further strengthened by the fact that the estimated lifespan of the erythrocyte is 4 months. So basically, the infant is born and they're protected from sickling. They have red cells that will last 4 months. And then the fetal cells will wear out, the new adult cells will come on board. And then she writes, this fits in well, both with Trout's observation that the fetal hemoglobin disappears at 4.5 months, and with our finding of an infant who developed 90% sickling at 4.5 months in comparison with 6% sickling at birth. So she basically, based on careful clinical observation and inference, figured out that fetal hemoglobin was the main modifier of sickle cell disease. In the years to come, sickle cell anemia was described as a molecular disease. The mutations were identified, and we understood more and more how the hemoglobin gene, the beta globin gene that's mutant in sickle cell disease, beta S, is in a cluster of genes that are developmentally regulated, including the gamma globin genes that make fetal hemoglobin. And these are the main output of the locus prior to birth, that we all rely on fetal hemoglobin to sustain us in the womb. And then after birth, in the transcriptional switch, fetal globin gets turned off, adult globin gets turned on. And for individuals with sickle cell disease who carry mutations in sickle hemoglobin, that's when their disease begins to manifest in infancy. And so a longstanding therapeutic goal, spurred on by observations like Janet Watson made, was, could we understand this well enough to reverse the switch and turn back on ameliorating fetal hemoglobin? So I got introduced to this problem when I was a hematology fellow working in Stu Orkin's lab at Boston Children's Hospital. And at the time, it was an era where recently genome-wide association studies had been done. Common genetic variants have been identified that were associated with the clinical severity of sickle cell disease. Like many, GWAS signals, these variants were not in the coding sequences, but were in non-coding sequences, and it was unclear how they worked. And then as the maps of chromatin and different cell types were beginning to become available, we observed there was a signature of open chromatin in erythroid precursors overlapping the genetic variants, suggesting a mechanism for this trait association where common genetic variation might affect the regulation of BCL11A. And BCL11A in turn had been shown to be the main repressor of fetal globin. And with time, we've learned BCL11A actually binds and represses the fetal globin gene in the adult state. So we were interested in understanding how important these genetic elements might be. And how could we do that? Well, we could do standard biochemical enhancer assays or gain of function experiments, but to really understand what a sequence does, it's in its endogenous context in the genome, we want to be able to perturb it. And so we turn to genome editing, where we can use molecular technologies to disrupt genetic sequences in situ. And at the time, it was kind of like the version 1 of the toolkit, where we had these gene editors that relied on protein as the DNA recognition molecule. And so we had talents where we could engineer them to bind to specific sites. And so we used talons and disrupted BCL11A enhancer ortholog in mouse erythroid cells. And what we observed was when we deleted these like 10,000 base pairs of sequences from the mouse erythroid cells, we saw almost complete loss of BCL11A, suggesting these sequences were totally required for BCL11A to be expressed. But when we deleted them from other cells that also expressed high levels of BCL11A, like B lymphocytes, there was no change in BCL11A, suggesting that this was really an exquisitely context-specific gene regulatory element. And that's become like a dogma of GWAS, that common genetic variants modulate these context-specific gene regulatory elements. And so at the time, Cas9 was being described as a gene editing system that could make genetic modifications in human cells. And this was, initially described as a prokaryotic immune defense system, but that could be easily repurposed just by swapping in a new guide RNA spacer sequence shown here in orange, where just with Watson-Crick base pair complementarity, the Cas9 nuclease could now bind to a new DNA target and create a double strand break, and then lead to genome editing effects through DNA repair. And so this is kind of an artistic depiction of Cas nuclease scanning a genome, finding these adjacent sequences. For the Cas9 strep pyogenes version, this is the NGG prospacer adjacent motif that the Cas9 first docks onto, and then unwinds its target with the guide RNA. With sequence complementarity, it's able to have a conformational change to engage its nuclease effector functions. And so because of this system's ease of reprogramming, it's scalable, and we could synthesize like an enhancer saturating library where we could target lots of perturbations to an enhancer element, let's say. And so we were interested in the BCL11A enhancer. And so we did this working with Feng Zhang at the time and Matt Canver, who was an MD.PhD student at the time. And what we observed were the results of each of these dots, which each dot is a guide RNA at the three main enhancers of BCL11A, the 62, 58, and 55, named by their distance from the transcription start site. And on the y-axis is the level of fetal hemoglobin enrichment. And what we saw were these stripes, especially this powerful stripe at the plus 58 element, where co-localized guide RNAs had a similar effect, where disrupting this narrow stretch of sequences would lead to loss of BCL11A expression and induction of fetal hemoglobin. And turns out the underlying sequences that are being disrupted are binding sites for the main erythroid transcription factor, GATA1 and tau1, which activate these sequences. And so now we had a simple therapeutic vision that we could use gene editing to disrupt minimal sequence elements of a single transcription factor binding site at the BCL11A erythroid enhancer. This would lead to selective loss of BCL11A in erythroid cells, but preserve BCL11A in non-erythroid contexts. And this could lead to high fetal hemoglobin in the red cell progeny. One could perform gene editing in autologous hematopoietic stem cells, use these as a graft, engraft the patient with his or her own gene-modified cells. And if a subset of cells were edited, in sickle cell disease, the fetal hemoglobin expressing red cells would have a survival advantage. So even a partial editing of hematopoietic stem cells could lead to an outsized benefit in terms of fetal hemoglobin expressing red blood cells. Working with our colleague, Scott Wolf at University of Massachusetts, my postdoctoral fellow, Yushun Wu, developed a method for highly efficient gene editing ex vivo of hematopoietic stem cells from sickle cell disease patients. So this is like a typical sequencing result where we read out the alleles after editing the BCL11A enhancer in the sickle cell disease patient hematopoietic stem and progenitor cells. And what we see here is like 2% of the alleles are unedited, 98% of the alleles are edited, and each of these edits disrupts the core GATA1 binding motif. These cells can be used for hematopoietic stem cell function, like engraftment in immunodeficient mice, and their red cell progeny are protected from deoxygenation-induced red cell shape change sickling. CRISPR Therapeutics and Vertex licensed the technology, developed the method, conducted clinical trials, and here are some of their results where they could see in individuals with sickle cell disease. Here's 43 individuals followed over time. Approximately 75% indels in the engrafting peripheral blood cells, about 40% fetal hemoglobin from a very low baseline. And each dot here is a vaso-occlusive crisis before the therapy. And then on the right, after the therapy, and you can see a great reduction in these painful crises, which are a hallmark and devastating feature of sickle cell disease. And so this led to the approval of Casgevy in December 2023 as treatment for sickle cell disease. Oh, great. Okay, so these trials led by Vertex CRISPR Therapeutics are one of these trials, but there's actually other approaches that are all based on the idea of Cas nuclease disrupting a genetic element, and then non-homologous end joining, repairing that double strand break and producing short insertions and deletions, and those can disrupt genetic elements. And if those elements are repressing fetal hemoglobin, then the result may be to induce fetal hemoglobin. And so there's also a trial by Bauer laboratories in China using Cas9 for TDT, which is transfusion-dependent thalassemia, also targeting the B7 enhancer. Editas has a trial for sickle cell disease using Cas12a, a different nuclease targeting the gamma globin promoter. In fact, the sequences where B7a binds. And then there's been a number of other trials that have since been suspended for various reasons. Shayo, do you want me to save questions for the end or do them in real time?
Yeah. Let's save them till the end.
Okay, so when you make a double strand break, you can actually harness that in another way. And the other way is through homology-directed repair. And so if there's an extra chromosomal DNA template that can be swapped in, and one can actually precisely repair a mutation. That's very appealing for genetic disease. You could presumably have the most parsimonious therapy would be to correct the mutation itself. But there's some challenges of this approach. It requires this extra chromosomal donor template sequence to be present in high levels. That requires some challenges like viral delivery to the cells. And importantly, this repair pathway is only active in cycling cells. So it requires some ex vivo manipulation to get cells, which are by nature quiescent stem cells, into the cycle to be able to do this repair. And it's competing. So if you have a double strand break, you might repair some with non-homologous end joining and others with homology-directed repair. And for example, at the sickle cell mutation site in beta globin, if you create indels, you might actually convert a sickle cell allele to a thalassemia allele. So this is a kind of harder approach at some levels, but there's a trial that was sponsored by Graphite Bio that's since been suspended. There was a severe adverse event of pancytopenia that was deemed likely related to study treatment, and actually that program has been closed. But then there's an academic group at Berkeley who's taking forward a trial, and that's ongoing. There's other forms of gene editing. As you may know, base editing is a form that can produce predominantly transition mutations. So here, it's Cas9, but an attenuated Cas9 that makes a neck rather than a double strand break, and it can be fused to a deaminase. And so like a cytidine deaminase or adenosine deaminase can make a base edit like a C to a U or an A to an I, and then the downstream DNA repair can resolve it like from U to T. So ultimately you can make a C to T change or an A to G change using base editing. And so how could this be harnessed for sickle cell disease? Well, in a couple ways. One is it could be used for disruption of an element, another for repair. And in fact, my postdoctoral fellow Jing Zhang, working with our colleague Keith Jung, showed that if we perform base edits targeting the same GATA1 binding site as we would target with the indels using nuclease, we could see a similar induction of fetal hemoglobin. So one could disrupt the B7A enhancer very effectively using base editing as well as using nuclease editing, and also see benefit in terms of fetal hemoglobin induction and prevention of sickling. Another approach taken forward by David Liu and colleagues has been targeting the mutation directly, the codon that encodes for valine that causes sickle cell disease. Now, you can't precisely repair that to glutamic acid, but you can convert it from valine to alanine, because you can't make the A to T transversion, but you can make the A to G transition mutation. And that is a hemoglobin variant called hemoglobin G Macassar, and that variant is benign and it doesn't cause sickle cell disease. So you could potentially convert from a very devastating hemoglobin disorder to a benign one. And they've shown, really nice editing and expression of this variant hemoglobin and prevention of sickling. And then there's even newer systems like prime editing. This is a flexible system where the attenuated Cas9, in this case, next to the other strand, there's an extended guide RNA that includes a template sequence. There's a reverse transcriptase, which can bind to the liberated flap, and then using the template sequence, write in new DNA sequences flexibly. So basically the red might be the new DNA sequence, which might be repairing the mutation or whatever the desired edit would be. And then this 3' DNA flap can equilibrate with the pre-existing 5' flap and now introduce an edit. Mismatch repair might see that as a mismatch and revert it. But if one evades mismatch repair, one can permanently incorporate that edit. So that's a very versatile gene editing system. And again, David Liu and colleagues showed that this could be used in this case to actually repair the sickle cell mutation from the A to T. And in this case, they had really excellent results with prime editing, where they could express normal beta globin in sickle cell disease patient cells and prevent sickling. So we now have this kind of abundance of riches where there's so many tools. We have lentivirus, we have zinc finger talons, Cas nuclease, base editor, prime editor. And sickle cell disease, there's actually like 3 distinct goals we could try to achieve to fix the underlying problem. One could be to add a globin gene, and this is the approach that Bluebird Bio has taken forward and also got an FDA approval in December of 2023. And here, using a lentivirus, you could add a non-sickling globin gene and thus interfere with the sickling process and mitigate the problems of sickle cell disease. One could induce the pyrologous fetal gamma globin using a variety of approaches like an RNAi therapy knocking down B7A, and there's an investigator-initiated trial at Boston Children's Hospital that uses a lentivirus to deliver that kind of an RNA. You can have B7A enhancer editing, which is what Casgevy relies on. You could recreate these rare conditions of hereditary persistence of fetal hemoglobin, which is kind of the Editas approach to target the gamma globin promoter itself. And then one could repair the beta globin with homology repair, with a base edit to make a variant hemoglobin, or prime editing to make the normal hemoglobin. So how do we actually implement that? So I touched on this at the beginning, but basically it starts with finding a patient who would be an appropriate subject for this therapy, and then asking if they want to participate. And if so, one could mobilize, which means giving a medicine that makes the hematopoietic stem cells, which normally reside in the bone marrow, temporarily move into the blood and then collect them from the blood. And then outside the body, these collected cells, which are heterogeneous, which contain rare stem cells and common progenitor cells, could be modified in some way by gene therapy, whether it be with a lentivirus or a nuclease or a base editor or so forth. And then these genetically modified cells can be used back into the blood of the patient. They can home back into the bone marrow. But to effectively engraft the patient has to receive myeloablative chemotherapy. So this is chemotherapy that basically wipes out the blood system to allow the new blood cells to take hold and reestablish a blood system. Now one can follow the patient in terms of their hematology. Have they had a benefit in terms of the hemoglobins that are produced? And then most importantly, have they had clinical benefits? And so what are the main bottlenecks here? I'd say the main two bottlenecks currently to allowing more wide access to these kinds of therapies are the requirement for ex vivo cell manipulation. So the potential solution to that problem could be a therapy that could be delivered in vivo. The other bottleneck is the requirement for the intense chemotherapy to replace the blood system. And if we could have non-genotoxic or less toxic ways to do the preparative conditioning therapy, that would also greatly simplify and make safer these therapies. So, you know, we're kind of faced with this dilemma of several good options now. So how do we choose among them? And here are some of the criteria I think we might consider. One is the efficiency, which could be at the level of cells or at the level of alleles. Are the cells modified? Are the alleles modified? You know, and that may be different for each disease, what's really needed to get a good effect or a great effect. The potency, you know, yes, you made a modification, but how beneficial is that modification? The precision, are those modifications perfectly what you intended? And if not, does it matter? Are they undesirable in it? And why would they be undesirable? The specificity, is there any other effects throughout the genome? And are any of those effects worrisome or adverse? The delivery, how do you actually get the modifications into the hematopoietic stem cells where they're needed? And the complexity, and complex approaches are really hard to deliver, and we need simple approaches to be able to scale them. So we don't really know yet how to choose, and I think we'll need close clinical follow-up, and we have a number of therapies being tested or just coming to market, and I think we'll need time to closely follow our patients and learn from them, and kind of help us improve and choose going forward. So I have a few examples of some of these concepts. So like in terms of efficiency, there's different ways you could measure that. So it could be like, how many modifications are there per engrafting clone? So in the Bluebird Bio product, which is called Beta Cell, and this was for transfusion-dependent thalassemia, but essentially the same product is also now approved for sickle cell disease. They were able to show that the drug product efficiency of transduction was associated with the clinical outcome, which was transfusion independent. So on the middle, you can see these kind of orange dots were patients who got transduced cells in the kind of 50–60% range, and those patients were not transfusion independent. They still required transfusions, whereas the blue were the other ones who became transfusion independent. And for the most part, they were greater than 62%, let's say, and had a higher transduction frequency, although there were some here who kind of had a intermediate transduction efficiency, but still had a seemingly good clinical outcome. So the predictors aren't perfect, but they're pretty good. Another could be not the ratio, but the absolute number of engrafting clones. And here is kind of a cautionary tale where the Graphite Bio had a homology-directed repair approach to directly repair the sickle cell mutation using Cas9 gene editing, and they observed in their first patient prolonged low blood counts, pancytopenia, after the therapy requiring transfusion and growth factor support. And as a result, I decided to stop that program. And it seems, although, the data is still being collected, that the problem is there weren't enough engrafting clones to replace the blood system. Another question is the potency. And so how much like fetal hemoglobin do we need to cure sickle cell disease? And that's a hard question. And like when we ask cure sickle cell disease, how do we define cure for sickle cell disease? So I think one way we might consider a cure would be long-term outcomes that are at least as good as seen in carriers of sickle cell trait, those with hemoglobin AS, one normal and one sickle cell allele. And so how much fetal hemoglobin do we need to get that? Well, here's a series from 13 children in Brazil who had compound heterozygous hemoglobin S, hereditary persistence of fetal hemoglobin. You can see the fetal hemoglobin here is like 43, 41%. So pretty similar to what's been reported like in the Vertex clinical trial participants. And they have few but non-zero pain crises, few but non-zero acute chest syndrome, few but non-zero transfusions. So it may be that this is a very good level of fetal hemoglobin that can prevent a lot of problems, but maybe it's not quite there. And if you look back at kind of older data predicting how much fetal hemoglobin per fetal hemoglobin containing red cell is required to prevent sickle hemoglobin polymer formation, it's approximately 15 picograms, which is approximately 50% of the hemoglobin in the cell. And here's another way of looking at it based on a calculation of the amount of non-cycling hemoglobin required to prevent polymerization at a given oxygen tension. And you can see here shaded is the 65% non-sickle hemoglobin that's present in individuals with sickle cell trait. And to achieve a similar degree of anti-sickling, you probably need 50% fetal hemoglobin. And let's say the trials to date, or those patients from Brazil I reported, have a little bit lower. So maybe we're close, but maybe not quite there yet. And so some work in my lab, we're investigating ways to get even more fetal hemoglobin to get even better clinical outcomes, we would surmise. And here, B7A has multiple erythroid enhancers. And if we target 2 enhancers simultaneously with gene editing, we can get more fetal hemoglobin than for any of the comparative approaches like knocking down B7A or editing gamma globin or just a single B7A enhancer. One of the main questions that comes up for gene editing is the specificity. Are there off-target effects? Are there other places in the genome that are modified that are not the target locus? Let's say not the B7A enhancer for the case of Casgevy. Well, we took the approach that although we could look at predicted off targets using a human reference genome, or we could test it in a handful of donors, that was really limiting our analysis to one genome or a handful of genomes. And we know there's lots of genomic diversity amongst all of us. And so we wanted to be able to look more broadly to encompass genetic variation. And so with our computational colleagues shown here on the right, developed a tool called CRISPR-Me to predict off-target effects that might occur not just due to the similar sites in the reference genome, but also influenced by genetic variants. And what we predicted was the top off-target site for the B7A enhancer targeting guide RNA was this one pointed to by this arrow. And so what does that SNP do? On the bottom strand here, you can see TGC is the reference genotype, and then TGG is what the SNP changes that C to a G. So now it creates a PAM sequence for the Cas9, a recognition sequence for Cas9, and now there's just 3 mismatches. So it turns what Cas9 can't recognize into something that Cas9 can easily recognize. So how frequent is this SNP? Actually, it's relatively common, and it's especially common in African or African ancestry individuals, where it's about 4.5% allele frequency. So now we have a therapy for sickle cell disease, which predominantly affects individuals of African ancestry, that could have its specificity modified by a common genetic variant that's particularly prevalent in individuals of African ancestry. So does that actually cause gene editing? We were lucky enough to kind of find an individual heterozygous for this variant where we could test the effects of gene editing. And what we found was, indeed, there was gene editing at this off-target site. This off-target site's on the Q arm of chromosome 2, 150 megabases away from B7A, which is on the P arm of chromosome 2. And so we hypothesized if there was simultaneous cleavage at both the on-target and the genetic variant-associated off-target sequence, then now that 150 megabases could become a pericentric inversion, and you could actually have a chromosome scale change that could be read out simply by PCR using primers specific for those inversions. And sure enough, we showed in hematopoietic stem progenitor cells that this happened at about a 1 in 600 allele frequency. And so this is a computational tool. It's widely available. I think there's a generic risk of any gene modification approach that it could be influenced by genetic variation. It's a challenging problem, but I think it's somewhat tractable, and there's now methods to look at it. Now, whether these off-targets have functional consequence, I think we'll only know by carefully following patients, and I hope and expect that they well may not have functional consequence. How do we deliver this better and avoid chemotherapy? Well, a lot of people are working on a lot of different clever strategies for this. So I'm optimistic we can get there. Here's one approach, using antibodies that are coupled to drug conjugates. So this is like a CD45 antibody drug conjugate that we tested with our collaborators, John Tisdale and Salome Demirci at the National Heart Lung Blood Institute, where they have a model of autologous transplant in the rhesus macaque non-human primate, where it's very similar to a clinical protocol design where you can mobilize this hematopoietic stem progenitor cells, edit them at B7A, the way we might do for a patient with sickle cell disease. Then after giving conditioning therapy, engraft them and then follow the animals over time. And in gray are animals that got conventional chemotherapy, and we saw high levels of edits in the peripheral blood over weeks and years. And then the colors are animals that we gave the antibody to. We didn't give conventional chemotherapy to. The blue animal got a low dose, and you can see kind of stabilize around 60% editing, and the green and the red got a higher dose that was well tolerated and stabilized at very high levels, like 85, 90% editing, similar to the chemotherapy treated animals. And the fetal hemoglobin, we see this kind of two waves, an early, very high wave during reconstitution, recovery from the therapy, and a later stabilization. And we see stabilization at a similar high level in the animals that got the antibody or the chemotherapy. So to conclude, I think we have a lot of opportunity in sickle cell disease genome editing and blood disorder genome editing, because we can access the blood system, we can modify cells outside the body, but hopefully we can even someday modify cells in situ. We can follow patients for a long time, and because blood cells are accessible, we can understand what we've done at a molecular, cellular, and clinical level, and hopefully have an impact on unmet clinical need. So with that, I'll stop. I'll thank people who contributed to the work, colleagues, collaborators, funders, and really patients who've inspired the studies.
All right, Dan, thank you so much for a fascinating talk. I think you did a great job at sort of overviewing from the very beginning in this discovery of fetal hemoglobin and how it's regulated all the way to a therapy based on that, and highlighting the future of what's to come in this field. We have a number of questions. I'm going to try and speed through them quickly so we can get to the second part, which is our patient interview. We're excited to have Faiza join us. Okay, so the first question was related to the patients who were essentially incomplete responders, so still had vaso-occlusive crises even after the gene editing treatment. Could you speak to why that might be?
Yeah, I think it's a great question. I think we really don't know yet, and I think we'll need a lot of follow-up to understand that. One possibility is that the level of fetal hemoglobin just wasn't high enough, whether it's per cell or the total number of cells to protect. But, you know, sickle cell disease patients who've received this therapy have experienced, a lifetime of sickle cell disease up to the point of the therapy, and they have a complex medical condition. And so whether all of the damage that's been done by sickle cell disease is reversible is also something that we really don't know yet, and we'll learn with time. So I think we know that in those patients, the hematology looks excellent in the sense that there's much higher hemoglobin, there's much lower turnover of the red blood cells, but it's not totally normal. So whether it's incomplete hematologic response or just non-hematologic reasons, I think we don't know yet.
Gotcha. And I guess on the point of maybe the editing strategy not leading to sufficient response, another question we had was whether or not there'd be room for a better BCL11A targeted strategy for sickle cell disease or thalassemia, or could there be a potential that treating the diseases at their root and like inserting a functional copy of beta globin could be favorable to BCL11A knockout, and what sort of bar would need to be reached for that insertion strategy, that replacement strategy, to really be selected over these current BCL11A knockout strategies?
Yeah, I mean, again, I think we need clinical follow-up. So like, I don't think anyone can answer it. And if someone, says a answer, like I would be skeptical. My personal view is, and I showed some data that certainly you can target B7A in other ways and get more fetal hemoglobin. I think there will be thresholds of fetal hemoglobin that do prevent all the hematologic problems of sickle cell disease. I think for the gene repair or gene addition strategies, the gene addition strategies tend to be somewhat heterogeneous in terms of how many copies are in the cell and where those copies are in the cell. And so I think by the current technology, they're not likely to give extremely uniform results that every cell will have the same outcome. And the repair strategies to date have also been largely less efficient. So then you have a population of cells where some are corrected and some are not. So but I think if we could repair every allele in every cell, certainly that would fix the problem.
Got it. And one last question. Sorry that we're not able to get to everything. I just want to make sure we have time for the next part of the session. So we got a couple questions around monitoring for off-target effects and the extent to which patients are monitored, either sequencing to assess for those issues. One person asking whether or not there's a future for sort of whole genome sequencing of patients to assess for potential off targets prior to the administration of therapy, and also a question about what it currently looks like today.
Yeah, I think that those are great questions. I think what it currently looks like today is largely a preclinical assessment, meaning the sponsor who brings the kind of innovative therapy, investigational therapy forward, does a thorough preclinical assessment, looks for off-target effects. If they're not present, great. If they are present, does a risk assessment to see, are they risky or not? And if the overall risk benefit favors moving forward, then it moves forward. I think we will move to an era where if there are like genetic variant associated off targets and we can know about them and screen for them, that we will test for them and see if they have an impact. But I'm hopeful with kind of better prediction, we can exonerate a lot of them. If you make a tiny genetic change at somewhere in the genome, the vast majority of the time, it's not likely to be functional. So, but we just don't have great ways to predict that today. In terms of the whole genome sequence, it's kind of back to that issue of more information than we know how to handle. So we could do it, but how to use it and how that impacts the ability to actually deliver the therapy to the patient in a reasonable amount of time, we don't really have those methods now, but I'm hopeful we could develop them in the future.
Gotcha. Okay. All right. Thank you for that. Okay, I think we're ready to move on to the next part of the session. This is sort of new for us as Translate, but on top of getting a physician perspective, we really also wanted to bring a patient perspective in for this second part of the session. So we're joined by Faiza, who is a patient with sickle cell disease who was treated using gene therapy, and really looking forward to learning a little bit more about her experience and sort of seeing the real life version of all of what Dan just brought us through. So I'll pass it to Jessica to get us started, and thank you so much to Faiza for joining us.
Yeah, thank you for the intro, and thank you again, Faiza, for joining us. If you want to just start by introducing yourself, and then we can get into the questions.
Hi, my name is Faiza. Yeah, I don't know what else to say, but
Yeah.
I'm happy to be here though.
Okay, sounds great. We're happy to have you. I think one question to just kind of start things off the bat, what was life like before the therapy, and like what were the changes that you feel like you've seen, just kind of starting off the bat?
Some of my changes that I mean, actually, my life before, I would say I had a lot of inconsistency. There was a lot of inconsistency in terms of social life, work life. Even before that, I was majority of the time like in the hospital. Now to give you a background, I am from Uganda, and I came here when I was about 11. When I was in Uganda, like I would have pain. I didn't really know that I had sickle cells. Probably my parents did know. I was diagnosed at the age of 3. And I would go from time to time, I'll go see my hematologist. But I did not really grasp the idea that I had sickle cells until when I came here. But I didn't start in Boston per se. I started living in Chicago. And the first crisis that I had to go to the hospital, and they basically told me, "Oh, describe your pain." And that was the first time I was asked to describe my pain. I'm like, I don't know. I'm just feeling a lot of chest pain. And I think from that point on, that's when I started to realize, okay, I have a condition called sickle cells. And from time to time, that's every single crisis that happened. That's when I learned that I actually have a disease. And we then moved to Kentucky. I had a few crises there. When we moved to Kentucky, that's when my mom made it a point that we're moving to Kentucky so that your health can be better. Maybe perhaps Chicago is too cold for you. Let's move to a southern state. So that's when I'm learning more until I came to Boston and I met Dr. Natasha. She explained to me what sickle cell was, and I started to become more aware and how to describe my pain and everything like that. So it was a learning process, to put it that way, before. And I learned how to adjust to it, what can trigger it. Yeah, I was just learning in the process. Let me put it that way.
Yeah, no, that is super helpful to kind of keep in mind, especially with something like this. So I guess along the way, what did education look like for you in terms of how people told you about, what these pains could indicate, whether that was coming from someone in the healthcare team or just from things you learned on your own?
Okay. So I asked my mom, like, how do kids like back home look like with sickle cells? She told me some of them have like, they are malnutritious. So they do not look, because every time I would tell someone like, oh, I have sickle cells, they'll be like, oh, really? You don't look like it. And I was like, what do you mean I don't look like it? And that was because majority of the kids back home, they really look sick. But I guess in my case, I did not look quote unquote sick. A lot of people or kids that were dying at an early stage. So that got me to be concerned about myself. I'm like, okay, next birthday is here. Okay, am I gonna make it for the next birthday? So I was concerned about that. I was concerned about death. How am I gonna die? Am I gonna die in the hospital every single time that I was having sickle cell pain? Also, Dr. Natasha did a very good illustration because I'm a visual learner. So she would explain, okay, this is how the cell looks like. This is how your cells look. This is what is happening. Your basically your blood, like she would explain it in like easy terms for me. Like your blood is basically clotting up in one area. You're not having a lot of blood flow. So then I'll go back to my friends and they're like, oh, why are you in the hospital? And then I'll explain the same thing. Yeah, so it was kind of like back and forth. I'm learning and then I go back and also teach others. So it was helpful in that way. And every crisis I learned, okay, these are the medications, this is the protocol. I would ask Dr. Natasha, when should I come into the hospital? Because there were some, this is when I also had to learn that, okay, this is the period for me to go to the hospital. This is when I need attention. This is when I can handle it better at home. My mom, on the other hand, she would reach out to a lot of people, a lot of doctors at home. And they would tell her, I'll give her a lot of vegetables, let her stay hydrated, warm drinks. So that's what I was on. Yeah.
Got it. I'm glad you had almost like a force of people from multiple angles giving you different perspectives. Considering all of this and considering your journey to kind of both realizing yourself kind of what this meant and then working through your family and your healthcare team with it, what led you to consider gene therapy as a treatment option in the first place? And what was that decision-making process like for you?
So I wouldn't say it was surely by me when it came to the decision of making this decision. It was a group of, well, it started out with my mom. My mom wanted me to be cured. She was like, Pfizer, no, I'm seeing a lot of research that is being made. Let's try bone marrow therapy. She kept on telling Dr. Natasha, let's try bone marrow therapy, let's try it. At first I was like, no, I don't want to go through it. It seems like a very tough situation to be in, people going into my bones. And then, so I didn't understand what bone marrow therapy really meant. But my mom was very persistent about that. Then Dr. Natasha was like, okay, let's see what your siblings, if they can match you. I am a sister, an older sister to 3 young ones. So we are 4 and I'm the only one with the disease. The rest have traits. We did the matching, what do you call it? Bone marrow to see if I can match with them. But unfortunately, they only had 1 gene. So none of them could match me 100%. So then my mom was very devastated. And then she was working for somebody, and the person is called Debbie Langston or Deborah Langston. And Deborah Langston was an eye surgeon at MGH. So she knew that there are possibilities, that there is probably research about it. There is something that is there that can be helpful. And every single time I felt sick, my mom would express to her, oh, Pfizer is in the hospital this time. Pfizer is in the hospital this time. So she made the research and then she found out that, oh, there is a gene therapy that is happening in Boston. And I was like, by that time, actually, when it happened, I was at the hospital. I'm in pain. And my mom is like, Faisal, ask the doctors about the gene therapy. I'm like, Mommy, I'm in pain. She's like, no, ask them. And I was like, no, let's first hold on to it. It's not even a thing yet. And she's like, no, there are so many people that are doing it in Chicago, here, there. And I was like, okay. So once I left the hospital, that was in like 2018. Then I started getting interested into it. I started looking up other people that are doing it. There were some that were in UK and all of that. So eventually I reached out to Dr. Natasha. I asked Dr. Natasha if I could be a participant for it. And she at first was opposing it, but with more conversations, understanding what it meant for me, she said, okay, no problem. I could give you, I could share with you the contact and you can talk to them and see if it's still something that you want to go through it. So the very first appointment I did go with Debbie Langston because she was in the medical world. So I wanted her to be there with me in case I didn't understand something. She could probably translate it in an easier way for me. And that's how the journey began.
Wow, that again, it's a whole crew of people that
Yes.
and I think that's amazing. I guess so when did you exactly start taking the therapy? What did that timeline look like? You first kind of heard about it, started reaching out 2018, and since then, how has it come to here?
So in 2019, I think that's when things started getting serious. We started thinking about doing the stem cells collection. And when they briefed me, like I think I had like 3 sessions. I had questions, so they briefed me about any concerns that I had. They told me about. They answered my questions, and in 2019, I would say that's when we started doing the first collection, and we weren't successful. I remember with the first one, we weren't successful. We didn't get enough. So we thought, okay, we'll try again. But then COVID came around. When COVID came around, it paused everything for me. We waited until COVID kind of settled. So like around 20, the ending of 2020, that's when we came back up again. Collected the stem cells. I think the second time was also not successful. We tried the third time and it was successful. And that's when the journey began. When the journey began, I was scared of the unknown because I kept on telling my mom, one of my concerns that this is an experiment. I don't know what this means. Yes, the whole concept of it is great, but the journey of it is what I was scared for. And once they gave me the chemotherapy in January of 2021, and then that's when they gave me my stem cells, the genetically modified ones, and went through all of 2021 and 2022. It was the roughest time, but I am grateful for it. Yeah.
Could you tell us a little bit more about how you've been feeling since, now that we're in 2024 now, and what's changed in terms of both the day-to-day and your broad patterns?
Okay. I would say at first, actually, when the transition was made, I still had pain. And I was like, okay, this was supposed to work. Why am I still feeling pain? Why am I still going to the hospital? What's going on? So I started feeling down. That was like my lowest point. And then at the same time, I was going through a transition of chemotherapy that I was actually not mentally prepared for. It's a different story talking about it, that this is what's going to happen. This is what your body's going to do. It's going to go through this. But then it's different when you're actually going through it. So that transition was a bit harsh. I started becoming very hostile with my parents. She's like, calm down, calm down. She kept on telling my sister not to bug me so much as well. I was very, very hostile because I was like having, I think I had two crises within that year. But then the next year, things were getting better. And also within that year, you have to keep in mind, we're still transitioning from COVID, and there were still restrictions in terms of who can see you, what you can do, you can't go out. So I felt very much also alone because I was still in I had to be protected because I shouldn't be sick within that period. So I felt very depressed. And then as things progressed, my health started becoming better. I started seeing changes in me. The chemotherapy was weighing off as well in terms of the side effects. My energy started coming back up again. I went to school not feeling, I was not feeling as sick. Social-wise, I was getting better at it. Mental-wise, it was getting better. School, I was being consistent. So those were the things that I would say changed, and there was more consistency with everything in life. Yeah.
Got it. Thank you, Faiza, for sharing your story so far.
Yeah.
You mentioned something that I wanted to follow up on. Back right when you were deciding to receive the therapy, you mentioned that your doctor, Dr. Natasha, was originally against it. And I actually wanted to loop in Dan here as well, because I think it speaks towards how your doctor sort of counseled you through this process and what you were sort of what you were considering, what they were considering. And so maybe I'll jump to Dan first, and then I want to get your perspective too. But to Dan, how do you think about counseling patients like Pfizer or others that you've seen in terms of whether or not gene therapy is really the right option for them?
Yeah, I think it's pretty daunting. We have, something we call informed consent, where it's the process of describing a therapy and asking a patient if they want to participate in a therapy. And, part of that, we have a written document that has a lot of information that we go over and share with the patient. And it's a lot of information and a lot of overwhelming information about, life-threatening level side effects, percentages. How do you think about a percentage, unknowns where there isn't even a percentage? If there's a patient, there's just one outcome that the patient's going to have, but, you can say, well, we've seen this in other patients. So it's a big challenge to really achieve that. And I think it takes time, and different people learn in different ways, visually and otherwise. And so I think it's a process that doesn't happen like in one visit. I think, the other thing is when we provide that information, like we're not in the patient's shoes, and every patient's going to weigh that differently. And, some patients are gonna, we're gonna think this is perfect for you, and they're gonna say, no, we don't want it. And other patients, we say, no, you're not sick enough, quote unquote, to need it. And then they, like, how do we know, right? So, so I think it's a shared process of telling what we know, telling what we don't know, and kind of together exploring, what makes sense and what doesn't.
Yeah, it seems like for something like this, the education and information sharing part of it is really crucial. Faiza, I think you mentioned that you went through maybe 3 sessions where you're sort of the whole goal was just understanding what's going to happen. Could you say a little bit more about that? What was done in those sessions that really helped you get comfortable with what was going to happen through this therapeutic process?
Well, one, I had to go through surgery. I didn't know I was going to go through surgery within every session. And the surgery that was taking place was more in, oh my gosh, I forgot the area, but it was more around here. It's just every single time that I had to go through those, stem cell collection. I was given blood, one. And then the second thing is then I'll go in and they would collect. When they would collect pretty much like the whole day. So I would be there. It was cool seeing how they do things. I was asking questions and all of that. But the first one, when it did not happen, I was devastated. I was like, oh, am I going to make it? And then they were like, no, you're just needing a little bit more. So that when we tried it the second time around, I was still hopeful that, okay, maybe this time I would actually be successful with it. Unfortunately, that did not happen. So I kept on thinking, maybe it's me. So maybe I need to be eating something, maybe I don't know. It started becoming stressful in terms of like, would I still be a candidate? Let me put it that way, or would I not? But that was the only way that I could sorry, not the only way. That was when I was questioning myself whether I would still be successful with it or not. And it also I was having a few questions whether it would impact when they gave me back my genetically modified cells, if that would have an impact to it. But after talking to Dr. Ezrick, she said, no, it's just like my body was doing what it had to do and produced what it could produce at the moment.
Got it. Got it. Maybe a question, a follow-up to both of you and to Dan. So the process of collecting the cells seemed like it was a long one for you because you went through a couple collections that didn't succeed. And I wonder, to Dan, is that sort of emblematic or representative of what this process might actually look like for a lot of patients?
I think so. I think one of the things for sickle cell disease, the standard medicine that is used for stem cell collection is something called GCSF, and that's contraindicated in sickle cell disease and has been associated with severe adverse outcomes. And so when gene therapy was kind of beginning for sickle cell disease, more than 5 years ago, studies had to be done to test a different medicine called plerixafor, which is used, and it can work. But it's outside of sickle cell disease. It's usually used in combination with GCSF. So as a sole therapy, it's effective, but it's not the most effective at mobilizing stem cells. And then actually collecting the stem cells from the blood is a non-trivial process, and the nature of sickle cell disease actually changes the physical properties of how you do that collection and makes it even more challenging. So getting enough stem cells to do this sort of ex vivo outside the body gene modification often, like in Pfizer's case, requires multiple collections. So each collection might be a procedure to put a temporary catheter into a large vein, to then have sequential days of having the blood run through an instrument to collect the stem cells. And then after those few days, maybe the line would come out, and then the patient would wait and see the outcome, and then maybe need to come back a month later, and then a month after that. So that's it's very challenging, and patients are generally getting red blood cell transfusions during this time. And in sickle cell disease, red blood cell transfusions are even more complicated than they would be in other situations. And some patients have, it's hard to find appropriate blood to do that. And, that process from a patient saying, I want to participate, to actually getting to the point where they can get into the hospital to get the chemotherapy and get the cells, it could be 6 months, it could be almost close to a year. So that's a big challenge.
Yeah, it's definitely a tough process.
Sounds like one. I guess throwing it back to Pfizer for a bit, and Dan, feel free to jump in on this. Thinking about both now and then shifting kind of gears, looking towards the future, what would you say the day-to-day is like now? And would you recommend this process to other patients, starting with Pfizer?
Would I recommend it? I would recommend it, though I would advise a lot of support system because it is a tough decision from the very, very beginning of the process. For me, it was tough in terms of this is an experiment. This is not something that has not been well, in my world, it wasn't done before. It was new and everything. In terms of gene modification, and so it was cool at the same time, but it was very scary in what that could entail. But now, after going through it, I would definitely recommend it, though I would recommend it with a lot of support. And I would say they did give what is it, someone to speak to. So they did give counseling sessions when available, which was great because it made things a bit easier as well. So in terms of the future, I also see this as an open door to figuring out, because I was talking to Dr. Esrick and she told me that all of us have like different way the alpha and beta cells are for sickle cells. And she told me there were some of them that were for beta, some of them were for alpha. So it's interesting how all these genetic therapies are trying to give hope for each and every person that is being affected with sickle cells in different ways. And it also gives hope to those that are really going through pain. So definitely 100% would recommend it with a lot of support. Yeah.
Maybe a quick follow-up there. So in the time after the therapy, so after going through the chemo and the engraftment period and all of that, it seems like you've been sort of out of all of that for a little bit of time now. Do you feel that your sort of symptoms have improved and that the disease is sort of better than what it was before? Or what sort of differences are you able to tell there?
That is a very excellent question. So my mom is, the way she understood this therapy, she thought it's like a cure. And I kept on telling to her up to this day, Mommy, it's not a cure. I cannot say it is 100% a cure, right? Because we're still trying to see where this leads. I'm only in 3 years right now. So in the very, very beginning, we were all concerned. Why am I getting sick? We have just done it. Not understanding that the body does take process to shift the way things were to how things are going to be moving forward, right? So, well, ask the question one more time. I just lost track.
Yeah, I was wondering, like, what do your symptoms look like now? Do you still
Yes,
experience
yes.
pain crises? What does
Okay.
your clinical profile look
Yeah.
like?
So before, I was having crises. My crises have changed with time. Ever since I was young to actually when I became older, it was very interesting. Because when I first came, I was having a lot of chest pain. That was how my pain episodes were. A lot of chest pins and needles within the chest area. And then as time went on, I was in high school, transitioning to college time. I started having more back pain and then more leg pain. So it was the pain was shifting. And I was like, is this something normal? Is this something that's not normal? My legs would also start hurting. And I was like, Mommy, was this something that I was also having? I was back in Uganda. She's like, yeah. So knowing that this is how my pain was, and then to now after gene therapy, like I did mention, I did have 2 crises, and I was still having those type of pains, and my body was still shifting, and we're still not understanding that how the body shifts. But last year, very big progress. I had less pain. less to minimum pain. It would come just like, it would just be dull pain, but it wasn't like the type of pain that is starting to like, oh, maybe I need to go to the hospital or anything like that. So the pain has decreased. Even I say this in terms of pain of going to the hospital, like a crisis, that has 100% decreased. In terms of the pain that I usually feel before I get to the hospital or to the need of the hospital, that has also decreased from the time that I started gene therapy, or even before that. It has 100% or 80% decreased. So I think my body is still making progress through it all, to a point that sometimes I question myself, oh, I wonder how sickle cell pain looks like or feels like. So it is a good thing that I don't even recall how that is.
So
But I could, yeah. Mm-hmm.
yeah, no, I was just gonna say, it sounds like you're doing better, but I guess a lesson to be learned is that nothing is black and white. Like, it's not as though everything
Yeah.
appears, but it's a therapy that
Yes.
enables some improvement. I guess
Yeah.
a follow-up then to loop in Dan. Or sorry, did you have anything else to add there?
No.
Okay. So just to loop in Dan again, I guess getting to this question of quote unquote cures and like, or maybe a better way of framing is, what does it take to get to sort of the next level of efficacy, to the point where the word cure could even be brought into the conversation? What do you see as the sort of major bottlenecks to wider adoption of gene therapy and also improving the efficacy? You've sort of talked about it quite extensively during your talk, but I wonder if you could distill that down to your few key points on what you think the key limitations today are.
Yeah. Well, I mean, I really echo what Faiza said, that I'm really reluctant to call things a cure. I think foremost, we have to have humility, and a cure to me is something you look back on and you can say, after a lifetime of good health, that's a cure. So I think we need to uh, try to do the best we can, develop the best therapies we can, and hope for that, but acknowledge uncertainty. Sickle cell disease is just like among the most cruel diseases, I would say, in that it has these episodic, painful episodes, but it has insidious manifestations where all the organs are affected. And when patients, are of the age where they've been participating in these gene therapy trials, their body has been dealing with all those effects of sickle cell disease. So I think an ideal treatment for sickle cell disease could occur, before that happens. I think to reverse all that damage is a very difficult thing to do, right? So I think we need safer ways to deliver the therapy, ultimately to get past the need for the chemotherapy, which is so toxic, the ex vivo outside the body manipulation, which is so cumbersome, that we could deliver this in a way that could be simple and scalable for patients. And then, we're just all in it together, kind of being honest about what we know, what we don't know, and learning together, and hoping to achieve the very best outcomes.
Yeah. I guess on the topic of learning, I think an important question always to ask to Faiza and to any patient who we bring into these kind of conversations. Our audience is not all clinicians and clinical trainees, but a lot of us are. And I'm wondering if you could say something about, through the whole process, the physicians that sort of did the best for you and the clinicians you had the best experiences with through this long and challenging process. What sort of, I guess, wisdom could you give to our audience in terms of what was done right that we should sort of take away as something that we should do in our future careers? I know it's a big question, but
Yeah,
anything
it's a big question. But I would say they were honest. They were very, very honest. Honest in that even once I left the hospital, after the transition of the new stem cells, and I was home, I'm trying to go through the new diet, everything like that, trying to adjust. Dr. Esri called me and she's like, Pfizer, I'm sorry to tell you this news, but one of the gene therapy that has happened, there was another gene therapy that happened. Someone got cancer. I was like, and like, oh, I've just went through it. I'm trying to grasp everything, the new changes, and then you hit me with that. And she's like, oh, but we're going to make sure that, we're going to see if this has something to do with the gene therapy, has to do with, maybe it's just the person's body just reacting that way, but they were just honest with it. It took me a while. I was like, oh my gosh, I've just went through this just to get cancer. It does not make sense. But they were very, very, very honest. However, had the truth was, maybe the potentials were there, maybe the potentials weren't there. They were just honest throughout it all. And I appreciated that. The reality of things. Also, back to the situation of like, oh, is this a cure? They were very honest about it from the very jump. This is not a cure. We don't know what this entails. The body can backfire. The body can agree. The body can remain, decide to remain in the same state. So there were a lot of variables that could have happened with this situation, but I'm glad I'm on the better side of things.
Gotcha. Thank you for sharing that little piece of wisdom about honesty. I think is super important. Jessica, I know we need to go through the Q&A a little bit, but I'm just looking at the time and I realize we may not have time for that, unfortunately. So sorry to the questions that we didn't get to. But wanted to thank both Faiza and Dan for joining us. Your perspectives, Dan, on the science and Faiza on the patient experience are tremendously valuable, and we're very grateful to have you guys here. And this has been a great session. I hope you've all enjoyed it. We also wanted to let people know about our next session. So I'm hoping to put on one of our colleagues, Sean, who could talk a little bit about that. But again, thank you to Dan and Faiza for joining us.
Thank you guys so much.
Thank you for inviting.
Thank you.
So let me open this up here. So Sean, yeah, take it away.
Yeah, absolutely. And thanks again, Dr. Bauer and Faiza, for that really excellent presentation. I think this session was extremely topical for our next session coming up, where we are going to have Dr. Kiran Musunuru, who is a practicing cardiologist and professor of medicine, and also notably for his upcoming talk, one of the scientific co-founders and senior scientific advisors for Verve Therapeutics, which is working on doing gene editing for a much larger indication, in what some people might say is a much more indolent indication, hypercholesterolemia and hyperlipidemia. And so Dr. Musunuru is really sort of a pioneer in this space, did one of the first demonstrations of durable gene editing to durably reduce LDL-C and cholesterol in non-human primate models using base editing. And that idea was the basis for the company Verve Therapeutics, which many of you are probably familiar with, was in the news recently for going to a clinical trial and basically trying to demonstrate sort of a one-time shot to knock out PCSK9, which of course is the target of approved medications, inclisiran, evolocumab, and really sort of trying to demonstrate that we could, in a much larger indication like hypercholesterol, use the same gene editing approaches for patient benefit. So I'm super excited to see this. I mean, I would say, as any practicing physician, I have a lot of patients who are on statins and PCSK9 inhibitors. So super interesting to think about how the technologies that Dr. Bauer talked about today and that Dr. Faiza sort of talked about her own clinical experience, what it would look like to move that into much larger indications, but I think most notably indications where, again, we're thinking sort of about much more chronic therapies and sort of chronic conditions. So Dr. Musanur will have a lot to say about that, because like I said, he was sort of the scientific sort of founder of this company that's now thinking about doing this.
All right. Oh, sorry. I keep on messing up here. But so the QR code was up there. You may not have had a chance to scan it, but it'll also be in an email that we'll send in a follow-up. So thank you so much, everyone, for joining us. Thank you again, especially to Dan and to Faiza for joining us. We really appreciate it. And we hope to see you all at our next session. Thank you.
Thank you.